Professor Carafit here with a review of Chapter 32, How to Be Alive. There will be some content here that may not be in the chapter, but it's in the lecture. You'll be okay here. All right, so an introduction to anatomy and physiology, or form and function. So think of an animal like, imagine you're designing an animal. Imagine, we're robots, or you're building a robot. What kind of problems would a living thing face on the earth? So any real living thing or made up living thing would have to fight gravity. If it's on land, it would have to get oxygen. It would have to get fuel. It would have to get rid of waste. It would have to communicate. So the top of it would have to be able to talk to the bottom of it, and its legs would have to communicate with its brain and its arms. All of that kind of stuff are problems that an organism faces. So if you were designing an organism, you'd have to come up with systems to overcome those problems. And you can think about what some of those might be. And I ask, what constraints would limit these systems? This is kind of a very open-ended question, but from a biology standpoint, let's just talk about evolution. We're constrained by reality, right? We're not going to just grow brand new parts because we need them, for example. And we're not going to be able to be a mile tall, for example, because our bones would shatter if we weighed that much. So we're constrained by physics. We're constrained by biology in terms of what's possible. Now, there are lots of constraints. I'm just going to kind of have a vague look at a few of those here. Size and shape are constrained by evolutionary history. So the shape of an organism and the size of an organism. So organisms can only be so big because, once again, gravity and things like that. Also, shape and structures are constrained. What I mean by constrained is, for example, think of a bird and its wings. A bird doesn't have arms and wings. Its arms evolved into wings. So evolutionary structures tend to be, when you have something like wings evolve, they tend to be modified versions of older structures. So new structures evolve from old ones. You don't end up with an animal with wings and arms because in that case, those wings had to evolve from nowhere. There is no structure there that could become a wing. So once again, we're limited by our evolutionary past. We can really modify the organs we have or the genes we have. Size is a limited factor because we can only get so big. Scaling up doesn't work very well. And what we'll see is, because of common selective pressures and common constraints, we'll see convergence evolve in different organisms in different environments. So that kind of helps to drive that similar adaptation to similar problems. And something else that all organisms have to do is exchange things with the environment. Every cell needs to be close to the outside world. It has to be able to exchange gases and nutrients. It has to be able to get rid of waste, to bring in what it needs. And we can do that with direct exchange, as I said, or through the use of organ systems. So that's where we start studying how things are put together and how they work, to understand why things work the way they do, how structure and function is related. So the study of anatomy is the study of biological form and like the bones, the muscles, the tissues, so on and so forth. Whereas the study of physiology is the function that those organs and various structures perform. So anatomy is structure and physiology is function. But of course, form and function are closely tied together. You can look at an organ and often figure out what it does based on its shape. So if we look at single-celled organisms, kind of like the ones we looked at in the Protista chapter, so like this amoeba is a single-celled eukaryote, it exchanges things directly with the environment through its cell membrane. And simple animals like this cnidarian, it's in the phylum Cnidaria, this is called a hydra. It has a body plan that's only 2 cells thick. The inside of the organism has water in it. The outside of the organism has water in it. Those cells are still in direct contact with the lake that this thing's living in. They have direct contact with the environment. But things get more complicated when we start having many multicellular organisms here, so trillions of cells. Then cells in the middle of the organism cannot exchange with the outside world. Now we need organ systems in order to accomplish that. So what we see on this slide is just a model of any kind of multicellular organism. It could be a human or a whale or a duck-billed platypus or whatever. But here are the cells. There's tons of cells in here, and the cells are still bathed in fluid, kind of like the cells in the organisms we saw in the previous slide. But these cells are in a fluid called interstitial fluid. That's also known as tissue fluid. It's the fluid that your cells are surrounded by. Now that interstitial fluid exchanges items with the bloodstream. And in this case, this is our circulatory system that's moving items around the body. And what are those items? Well, the bloodstream picks up waste from cells. And it also deposits food and other things to cells. So that blood drops things off at the cells, picks up waste. That waste goes into the excretory system. And metabolic waste, that's waste produced by the cells, is lost as nitrogenous waste like urine. Meanwhile, the blood picks up nutrients from the digestive system, where food was brought in and broken down and absorbed and then left as waste. And then that blood picks up oxygen and releases CO2 and then drops it off with the cells. So in these cases, the cells are not in direct contact with the environment, but they are in direct contact with organ systems that are in contact with the environment. And those organ systems have structural adaptations that help us to overcome some of those limitations we've talked about. Lung tissue has lots and lots and lots of these air pockets, lots of surface. The kidneys have tons of these blood vessels wrapped around all over the place for getting rid of waste. Here we have the small intestines with lots and lots of projections that increase their ability to absorb nutrients. And we'll see that these kinds of adaptations, what we really saw in that previous slide was a lot of increase in surface area of various organs. And what we'll see is that other far distant organisms like plants and animals have both co-evolved, through convergence, somewhat similar looking adaptations to similar problems, because the same constraints are on the plants and the animals, the same pressures, and they sometimes come up with similar solutions. Here we see the xylem, the water transporting tissues of a plant, which are quite a bit similar to blood vessels in terms of their structure and function. We can see here a pea plant. This is a pea pod with lots of baby peas, right? These are young pea plants basically, and they're all hooked up to the mother, kind of like an umbilical cord. It kind of reminds you of these pigs all lined up and getting nutrition from their mom. These peas are all getting nutrition from the mother plant here. Similar kind of convergent idea. Here we see the tissue in our villi, our villi, that's the finger-like projections of our intestines that absorb things, and we can see little finger-like projections on a root of a plant. It increases the surface area, helps with absorption. Here's the gas exchange tissue in the leaf of a plant. Here's the tissue in a lung. We can see similar structures doing similar functions. So why is it that at least for some of these things, especially the bottom two, how could it be they arrive at such similar solutions? Well, one of the driving constraints on all of life is surface area to volume ratios. So that's surface area to volume. And what I mean by that is the ratio between surface area and volume. You could have surface area over volume, okay? So surface area is the amount of surface around an object. So let's pretend these are cells, okay? Here's a little one that's 1 millimeter wide on each side, and here's a bigger one that's 4 millimeters wide on each side, okay? And they're cube-shaped. Surface area is the amount of surface. That limits how much material can get in or out of the cell, because surface area is where things get in and out. It's the membrane of the cell in this case. It's kind of like the highways running into a city. That's how things get in and out. Volume is the space inside the cell, and volume determines how much material the cell needs, because that's where all the chemical reactions are happening. So how much volume a city has determines how many people it can hold, right? And they all need food and water, and the roads going into the city determine how much food and water can get in or out of that city. So you can think of those as kind of parallel ideas. Surface area is the amount of material that can get into a cell. Think of it like the highways running into a city. Well, the volume is how much determines how much material that cell or city requires. So it's a very delicate balance here. And the important thing to note, the last thing we want to talk about before we move on, is that they don't scale up in a 1-to-1 ratio. So if you have a little cell that has 1 unit, that could be a micrometer or a millimeter, it doesn't matter, but 1 unit big on each side of the cube, the surface area of a cube would be 6. And the volume, which is in a cubic unit, would be 1. So notice this is why it doesn't scale up right. Surface area contains squared units, whereas volume has cubic units. So 6 to 1, so 6 to 1 is a ratio of 6, right? If you double the size of that cell to units of 2 on each side of that cube, so 2 microns or 2 millimeters, you have a surface area of 24 and a volume of 8. And now when you do 24 over 8, you get a ratio of 3. That ratio has gone down all the way up to this point, where if you have a cell with 4 units wide, you only have a ratio of 1.5. So as we go from a little cell like this to a big cell like this one only has a surface area to volume ratio of 1.5, whereas this one had a ratio of 6. Meaning there was a whole lot of edge compared to the space inside, meaning it was easy to get enough stuff into this cell to keep it alive. But it's going to be very hard to get enough stuff into this cell and out of this cell to keep it alive, because the volume has increased out of proportion to the surface area. And that's an important concept for understanding the structure and function of a lot of our organ systems.